Hi everyone! In this video, we will be having a basic discussion about the central dogma of molecular biology.
The central dogma. The central dogma is a process by which the DNA instruction is converted into a functional product. In 1958, Francis Crick, who discovered the structure of DNA, proposed this central dogma. Talking to a molecular biology, central dogma explains the flow of genetic information from DNA to RNA to produce this functional product, which is protein. The central dogma suggests that the DNA contains the information needed to make all of our proteins, and the RNA is a messenger that carries this information to the ribosomes. The ribosome serves as a factories in a cell, wherein the information translated from a code into a functional product. The process by which the DNA instruction converted into a functional product is called gene expression, and this gene expression undergoes two stages, which is transcription and translation.
The DNA was discovered by Swiss biochemist Frederick Mistrer and later realized its central importance to biology. In eukaryotes, DNA is found inside the nucleus. It is tightly packed and is called chromosome. Made up of nucleotides, the building blocks made of phosphate group, a sugar group, and one of four types of nitrogen bases, adenine, thymine, guanine, and cytosine. It is ordered to determine what biological instructions are contained in the strand of the DNA. DNA contains instructions needed for an organism to develop, survive, and reproduce by converting sequences to messages. First, enzymes read the information in a DNA molecule and transcribe it into an intermediary molecule called mRNA. Second, the information contained in the mRNA is translated into language of amino acids. Furthermore, DNA double helix is termed to determine DNA's winding. Two-stranded chemical structures looks like a twisted ladder.
Now, let's talk about the RNA. RNA, or the ribonucleic acid, is a nucleic acid and single-stranded that function in a cellular protein synthesis. RNA is made up of chains of ribonucleotides, the nitrogenous bases bound to a ribose sugar, joined together by a post-covalent diester bands. The adenine, guanine, cytosine, and uracil, which replaced by the thymine in DNA, are the nitrogenous bases in RNA. The three most well-known and studied types of RNA are the messenger RNA or the mRNA, transfer RNA or the tRNA, and the ribosomal RNA or the rRNA, all of which are found in all organisms. These and other forms of RNA, including enzymes, mainly carry out biochemical reactions. The function of mRNA is to provide the following constituent codons that being read by the tRNA to produce specific protein. While then, the rRNA molecules is the major functional components of the molecular machine, the so-called ribosomes, which carry out the translation process.
Now, let's talk about the replication. DNA replication is a process that involves the production of identical DNA helices from one double-stranded molecule of DNA. Its overall process is extremely important for the cell growth, cell repair, and the reproduction of organisms. The first step of DNA replication is called the formation of replication fork. Before the DNA can be replicated, the double-stranded molecule must be unzipped into two strands. This unzipping is done by the enzyme called helicase. DNA helicase disrupts the hydrogen bonding between base pairs to separate the strands into Y-shaped form known as the replication fork. Replication fork is a bidirectional, meaning it is oriented in the 3' to 5' direction or known as the leading strand, while the other is the 5' to 3' direction or known as the lagging strand. This area will be the template for the replication to begin. The second step of DNA replication is called primer binding. The leading strand is the simplest to replicate. Once the DNA strands have been separated, a short piece of RNA called primer binds to the 3' end of the strand. The primer always binds to the strand as the starting point for replication. The primers are generated by the enzyme called DNA primase.
Since we are done discussing with the step 1 and step 2 in the DNA replication process, let us proceed to the step 3 elongation process. DNA polymerase is an enzyme responsible in creating new strand in replication process. There are 5 different known types DNA polymerases present in human and bacteria cell. In bacteria, the known polymerases are E. coli, polymerase 3, which is the main replication enzyme, and polymerases 1, 2, 4, and 5 are responsible in checking error and repair. Polymerase 3 will now bind to the site of the primer and begins adding new strand complementary to the strand during replication process. In eukaryotic cell, the DNA polymerases are alpha, delta, and epsilon. This is because the replication begins from 5' to 3' direction. The lagging of strand begins by binding to a multiple primers. Each primer is several bases apart. The last process of DNA replication is called termination. Exonuclease enzyme will remove all the primers in RNA. These primers then replace with appropriate bases. Exonuclease enzyme will now proofread the newly formed DNA to check, replace, and remove errors. DNA ligase will join the Okazaki fragments in forming single unified strand. Telomeres now act as a protective cap at the end of the chromosome. Telomeres will catalyze using an enzyme called telomerase. Once completed, parent strand and DNA coils to form double helix strand. Therefore, replication process produces two molecules, one from parent and one from new formed strand.
Together, let's learn on what is transcription. Transcription is a process by which the information in the DNA strand is being copied and transcribed in order to make a new RNA molecule. Transcription involves three processes. This includes the initiation stage, the elongation stage, and the termination stage. Transcription starts in the initiation stage where an enzyme called RNA polymerase and a number of accessory proteins called transcription factor is being bind into a specific DNA sequence called promoter. Together with the RNA polymerase and the transcription factor, it forms a transcription initiation complex which initiates the transcription process at the promoter. Then, it undergoes elongation stage, where the RNA polymerase is being synthesized using one of the DNA strand and adding new nucleotides or matching complementary bases to the strand. So once the strand is completely synthesized, it ends up signaling to the transcription process in the termination stage, and a newly RNA molecule is being formed.
After transcription, translation then proceeds. Translation is a process by which the genetic code contained within a messenger RNA molecule is decoded to produce a specific sequence of amino acids in a polypeptide chain. It occurs in the cytoplasm following transcription, and like transcription has three stages: the initiation, elongation, and termination. For translation to begin, the start codon AUG must be recognized. This is a codon specific to the amino acid methionine, which is nearly always the first amino acid in a polypeptide chain. At the 5' cap of mRNA, the small 40S subunit of the ribosome binds. Subsequently, the larger 60S subunit binds to complete the initiation complex. The next step, elongation, can now commence.
In elongation, the ribosome has two tRNA binding sites, the P site which holds the polypeptide chain and the A site which accepts tRNA. While methionine tRNA occupies the P site, the amino acyl tRNA that is complementary to the next codon binds with the A site using the energy yielded from the hydrolysis of JTP. The thionine moves from the P site to the A site to bind to the new amino acid there, and so the growth of the peptide has begun. The tRNA molecule in the P site no longer has an attached amino acid there, and so leaves the ribosome. The ribosome then translocates along the mRNA molecule to the next codon, again using energy yielded from the hydrolysis of the JTP. Now, the growing peptide lies at the P site, and the A site is open again for the binding of the next amino acid, and so the cycle continues. In termination, one of the three stop codons enters the A site. No tRNA molecules binds to this molecule, so the peptide and tRNA in the P site become hydrolyzed, releasing the polypeptide into the cytoplasm. The large and small subunits of the ribosome dissociates, ready for the next round of translation.
And since we have mentioned proteins, now I'm going to add basic information about the protein, specifically the structures. The identity of a protein can be identified with its structure. A protein is a structural unit that is composed of 4 structures, and these are the primary, secondary, tertiary, and the quaternary structures. The primary structure is the basic makeup of a protein. In here, you can see that the protein is composed of sequence of amino acids that are joined together or linked together by specific chemical bonding. Then we also have the secondary structure, which is composed of more than one chains of amino acids that are joined together by chemical bonding also. And next we have the tertiary structure, which is now considered as a protein molecule and is composed of one subunit. Myoglobin, for example, is a protein which is in a tertiary structure and is composed of one subunit. And lastly, we have the quaternary structure, which is composed of more than one subunits, and the example for that is the hemoglobin. Hemoglobin is the protein that can be found in the blood that is composed of 4 subunits and is in the quaternary structures. And those are just the basic information about the protein structure. And that concludes our discussion. Thanks for watching!